Welcome to my channel. In this video, we will be talking about another very important topic, that is endoplasmic reticulum. And at the end of this video, you will be having clear concept of rough endoplasmic reticulum and smooth endoplasmic reticulum and their function and their working as well. You will also get to know that how different organelles are linked with each other and they are basically helping each other's function. Well, in the previous video, we were talking about the nucleus, and we have explained that nucleus is a double membrane organelle, and this is the internal membrane. Here we can see the external membrane. Here we can see the nucleus pore. These are the nuclear pore, and here we can see the nucleolus. Now let's talk about endoplasmic reticulum. Endoplasmic reticulum is uh, also a double membrane organelle, and its membrane basically is extension of the nuclear outer membrane. The outer membrane of the nucleus, it extended and it folded again and again and again and again, and a structure is formed, and this structure is basically called as rough endoplasmic reticulum. Sometime the same outer nuclear membrane, it fold and it form channels, tubes like structure, and this tube like structure is basically called as smooth endoplasmic reticulum. On the rough endoplasmic reticulum, which look like a fold, and these fold are basically called as cisternae. This each fold, this fold, this fold, this fold, this fold, this fold, these folds are basically called as cisternae. Cisternae or cisternae, you can pronounce it as you want. On the rough endoplasmic reticulum, you can also see that ribosomes are also present. But on a smooth endoplasmic reticulum, which look like a channel-like structure, no ribosomes are present. And if we talk about the function of rough endoplasmic reticulum and smooth endoplasmic reticulum, due to the presence of ribosome, you must have get an idea that rough endoplasmic reticulum will be little bit related to the protein synthesis. And yes, it is related to the protein synthesis. Oh, now you can say that obviously the process of translation and protein synthesis happens in the ribosome. So how this endoplasmic reticulum, rough endoplasmic reticulum, is involved in the protein synthesis? Yes, you are right. Protein will be synthesized in the ribosomes. But after the synthesis, the immature protein, it will enter inside the endoplasmic reticulum, and some post translational modification will be taken place. Post translational modification mean the modification in the protein after the translation. So once the protein will be synthesized in the ribosomes, it will enter inside the endoplasmic reticulum, and after entry, some different kind of modification will be taken place on the on the protein in the endoplasmic reticulum. What type of these modification and how protein enter inside the endoplasmic reticulum will be discussed in this video. Stay with me till end of the video. I am very much sure that all your concepts related to smooth and rough endoplasmic reticulum will be clear. Now, first of all, let's see how the newly synthesized protein enter in the endoplasmic reticulum. So here you can see this is a ribosome, and a messenger RNA has been placed over it. And once this messenger RNA has been placed over it, the process of translation has been started, and here you can see the protein is being synthesized. Protein is being synthesized. But when the protein is being synthesized, the first 10 to 30 amino acid sequence, 10 to 30 amino acid sequence is basically called as signal peptide. Signal peptide. Now what is signal peptide? It's the first 10 to 30 amino acid sequence of every protein. Of every protein. Protein is made up of amino acid, na? So obviously the first 10 to 30 amino acids, they will be called as signal peptide. Why they are called as signal peptide? Because there is a protein that is called as signal recognition protein. Signal recognition protein. This protein basically recognize or can identify this signal peptide. this small proteins molecule, it can identify the initial segment of the protein, that is called as the signal peptide. This protein, this signal recognition protein, can identify it, and it can attach over it. It can attach over it. Let me draw it with the, yeah, here it is. So this signal recognition protein, it has recognized it, and it will attach over this signal peptide from one side, and other side it will attach with the ribosome. Okay, now this signal recognition protein will be having its receptor present on the endoplasmic reticulum. So this is the receptor for this signal recognition protein. So once this signal recognition protein has attached with the signal peptide, this complex, it will attach on the signal recognition protein receptor. Signal recognition protein receptor. Now this complex will attach over it, and once this complex will attach over it, this newly synthesized protein along with the signal peptide, it will be guided, it will be sent inside the lumen of endoplasmic reticulum through a channel that is called as translocon. Now here you can see that the protein, the newly synthesized protein, has been guided, has been sent, has been entered inside the lumen of the endoplasmic reticulum with the help of signal recognition protein and signal recognition protein receptor. Okay, now once this protein has entered inside the cell, now what will happen, the process of translation is still going on. Okay, and the protein will keep on elongating, and here you can see at the end there will be a signal signal peptide will be present, obviously. Now we can say that, for example, this is a complete protein that has been entered inside the endoplasmic reticulum. A complete protein after the synthesis from the ribosome has been entered inside the endoplasmic reticulum through translocane and by following this mechanism. Now what will happen inside the endoplasmic reticulum, there is another protein that is called as signal peptidase. So here you can see this is a signal peptidase. Now what will it do? It will just break this bond, and it will separate the signal peptide and rest of the protein. Now here is the signal peptide, and here you can see here is the rest of the protein. This is the protein, but you can see that this protein is linear protein. To get mature and to get active, protein must, this is basically called as primary structure of protein. This is basically primary structure of protein. You know that the linear structure is primary. This structure need to be folded. Now this structure need to be folded to convert into secondary and then tertiary and then quaternary protein. Now quaternary protein will be a little bit complex like this, and that will be mature. Now point is to convert from primary to secondary to tertiary to quaternary protein, glycolization or attachment of carbohydrate on this protein is also required. Now what will happen, some sugar groups or different carbohydrates moieties, they will be attached on the different parts of the protein, and these carbohydrates, they not only help in the folding of protein, they also have some other functions as well. Okay, so it means in the endoplasmic reticulum, two main things are happening. One, the signal peptide is being removed. Second, the primary protein that has been produced after the removal of signal peptide, it will be glycosylated. Glycosylated mean a carbohydrate or sugar moiety will be added. Glycosylation. Okay, now glycosylation will be done, and once the glycosylation will happen, obviously this linear protein, this primary protein, it will be folded to form secondary, tertiary and quaternary protein, and that quaternary protein is basically a mature protein. It's a mature protein. Now all these modification, the glycosylation and the folding and conversion of primary to quaternary protein, these are basically post translational modification, because they are happening after the translation. So anything that is happening after the translation will be post translational modification, and anything that was happening before translation, it was pre-translational. So these are all the post translational modification. So we can say that rough endoplasmic reticulum, not smooth, rough endoplasmic reticulum play very important role in the post translational modifications. Okay, now what will happen, this mature protein, it still needs some further modification, and those modification will be done in the Golgi apparatus. Obviously it's quaternary protein, but still it needs some modifications, and those further modification will not be done in the endoplasmic reticulum. Those further modification will be done in the Golgi apparatus. Now what will happen, this membrane of the endoplasmic reticulum, it will make a vesicle against this protein, and this vesicle, it will bud off, it will bud off in this way, and inside this bud there will be a quaternary protein. To some extent it's a mature protein, it contains a quaternary protein. Now this bud will remove from the endoplasmic reticulum having a quaternary protein. Now here you can see, let me show you, sorry, yeah, it's right there. Now for example, this is a bud and it is having a quaternary protein. Now with the help of cytoskeleton, you know, there are lots of proteins are scattered inside the cell, and they are forming a cytoskeleton. With the help of cytoskeleton, this protein, it will move toward the Golgi apparatus, and then this bud, it will fuse with the Golgi apparatus, and the quaternary protein, it will be transferred to the Golgi apparatus, where further post translational modification will be taken place. Now what will happen in the Golgi apparatus, we will discuss in the next video, because in today's video we were talking about the endoplasmic reticulum. Okay, so this was all about the endoplasmic reticulum, and here we have seen that endoplasmic reticulum is involved in the post translational modification. After the synthesis of protein will enter inside the endoplasmic reticulum, rough endoplasmic reticulum, signal peptide will be removed, a linear primary protein will be formed. Now this primary protein will be glycosylated, and then it will be converted into secondary and tertiary and quaternary protein. Quaternary protein is a kind of mature protein, obviously not mature enough, but it's still mature protein, and then this mature will be bud off, and it will move toward the Golgi apparatus, it will attach to it, and the protein will be released inside the Golgi apparatus, where further post translational modification will be taken place. Now in the next video we will see what will happen in the post translational modification. Now let's talk about another structure, that is smooth endoplasmic reticulum. Now let's see what will happen in the smooth endoplasmic reticulum. As we have said that as there are no ribosome, so obviously smooth endoplasmic reticulum has nothing to do with the translational or post translational modification. Clear? Okay, now here you can see smooth endoplasmic reticulum. This is the smooth endoplasmic reticulum. Here you can see this is the cell membrane, and smooth endoplasmic reticulum is present inside the cell. Now what happens when there is you are on starvation and there is low glucose level in your blood? You know what happened? The glycogen, the stored form of glucose, you know, in your hepatocytes and in your kidney cells and your muscle cell, the glucose was stored in the form of glycogen. Now what will happen? During starvation, when needed, when you are not taking glucose in, the stored glycogen, it will be converted into first glucose 1 phosphate, then glucose 6 phosphate inside the cytoplasm. This whole is the cytoplasm, and this is the smooth endoplasmic reticulum, this pink, and this whole is the cytoplasm outside. Now the glycogen first converted into glucose 1 phosphate, then glucose 6 phosphate. Now this glucose 6 phosphate will enter inside the smooth endoplasmic reticulum with the help of glucose 6 phosphate transporter. This is the glucose 6 phosphate transporter. Now once the glucose 6 phosphate has entered inside the cell, enter inside the smooth endoplasmic reticulum. Inside the smooth endoplasmic reticulum, here you can see glucose 6 phosphatase. Glucose 6 phosphatase. Now this glucose 6 phosphatase, it will convert the glucose 6 phosphate that has entered inside the smooth endoplasmic reticulum. It will convert glucose 6 phosphate into a separate phosphate and glucose and glucose. Okay, obviously at the sixth position, at the sixth carbon phosphate group was attached. This is the reason it was being called as the glucose 6 phosphate. This enzyme will remove that phosphate and the remaining entity will be called as the glucose. Now inside the smooth endoplasmic reticulum, glucose 6 phosphate has been converted into glucose. Now this glucose, with the help of glucose transporter 2, that is called as GLUT2, glucose transporter 2, it will come out of the smooth endoplasmic reticulum inside the cytoplasm, and from the cytoplasm, with the help of another GLUT, another glucose transporter 2, with the help of another glucose transporter that is present on the plasma membrane, it will cross this glucose transporter and it will enter inside the blood. Now it will distribute throughout the body to meet the energy demand. We know that when we need energy and we are not taking glucose from external source, the glycogen will be converted into glucose and it will enter inside the blood to meet our energy demand. So this is the first function of the smooth endoplasmic reticulum, present in the hepatocytes or in the tissue where the glycogen was stored, and this glycogen will be converted into glucose and enter inside the blood. So this is the first conversion, first use. The second use is inside the smooth endoplasmic reticulum, there are several other enzymes are present. And those enzymes are responsible for the synthesis of phospholipids. You know that phospholipids are required in the plasma membrane and in the membrane of organelles like nucleus and endoplasmic reticulum, and all these phospholipids are synthesized in the smooth endoplasmic reticulum. Okay, cholesterol is lipids, cholesterols is also synthesized in the smooth endoplasmic reticulum. It means cholesterol derivative hormones like estrogen, progesterone, androgens, they all are synthesized in the smooth endoplasmic reticulum of the cell. Okay, similarly steroids, mineralocorticoid and glucocorticoid, they are all these all steroids, they are synthesized in the smooth endoplasmic reticulum. Okay, different kind of enzymes are present. Furthermore, a big group of enzymes that is called as cytochrome P450 system. This system of enzymes is also present inside the smooth endoplasmic reticulum, and you know that this system basically responsible for the detoxification of certain toxins and drugs. This enzyme help in the detoxification of the certain drugs. Okay, so it means this is involved in the metabolism of certain toxins and compounds For example, if you take alcohol, and when your alcohol after getting absorbed, it enter into the liver, and in the liver it is get detoxified. So it means this enzyme is going to play a very important role in the detoxification of the toxins, and all these things happening in the smooth endoplasmic reticulum. Okay, now we also know that smooth endoplasmic reticulum is a reservoir, is a reservoir, is a storage house of a very important second messenger, that is calcium ion. It is a storage house of calcium. It is storage house of calcium. Now there are different mechanisms due to which this calcium come out of the endoplasmic smooth endoplasmic reticulum, and then it get absorbed. For example, we know that whenever a hormone or a drug, a substance, when it attaches to its receptor, a G protein coupled receptor, and this receptor, then it will activate uh, phospholipase C. Phospholipase C. Now this phospholipase C convert uh, a phospholipid that is called as PIP2, phosphatidol inositol diphosphate, and this phosphatidol inositol diphosphate, it will be converted into IP3 and DAG. Now this IP3 will be having its receptor on the smooth endoplasmic reticulum, and when this IP3 it will act on its receptor, the calcium channel will open and calcium will come out of the smooth endoplasmic reticulum inside the cytoplasm. Now it will attach with some other protein, calmodium, and calcium clomodium complex will be formed, and some other functions will be done. I have discussed detail of this cellular mechanism in my other video. Link will is given in the description. If you want to explore more about the role of calcium and role of IP2 and role of G protein coupled receptor, you can watch my video. Here I just want to let you know the function of smooth endoplasmic reticulum. Okay, now calcium will come out of the smooth endoplasmic reticulum, and it will perform certain function. If you want to see what are these functions, you can watch my other videos. Okay, now once the function has been done, here you can see the calcium is out of the smooth endoplasmic reticulum inside the cell. Now once the function has been done, this calcium has performed its function, this calcium will be up reuptaken. It will be reuptaken back into the smooth endoplasmic reticulum with the help of a transporter, a pump that is called as SERCA. Smooth endoplasmic reticulum calcium uh, active pump. Okay, now with the help of this pump, the calcium will move inside the smooth endoplasmic reticulum, back inside the smooth endoplasmic reticulum, and this pump will use ATP as well. Okay, so it means this is the energy dependent process. So in this way, smooth endoplasmic reticulum is also involved in regulating lots of function: synthesizing of hormone, detoxification of toxins, synthesizing of certain phospholipids, steroids, regulating calcium level inside the cell, and maintaining calcium dependent functions. So this was all about the smooth endoplasmic and rough endoplasmic reticulum. I hope you must like the video. Please don't forget to like, share, and subscribe, and press the bell icon as well. Thank you so much.